my name is Alekhya Kotha. I'm a junior at Triangle Math and Science Academy in Cary, North Carolina, and today I will be presenting about one very specific neurodegenerative disease. My project specifically revolves around the analysis on the downregulation of genes in epilepsy and the mRNA expression profiles of glioma-related epilepsy to develop a treatment that will ultimately improve the medications currently on the market in terms of effectiveness and also lessening side effects. This is a project involving computational biology, so it uses an analysis that I will be walking you through. So first thing is the background. Epilepsy is a very common neurological disorder associated with stigma and high psychiatric comorbidity, meaning that it is associated with various different disorders that can be hard to target all at once. Due to its complexity and effect on varying parts of the brain, it is extremely difficult to understand, even with more than 100,000 cases in North Carolina itself. Some symptoms of epilepsy include fainting or constant fatigue, muscle contractions or spasms, migraines, staring with or without muscle contractions, and stiffening of the body. In order to understand some mechanisms of epilepsy to create a targeted treatment, it is important to know that existing research has shown there are expression profiles of microRNA or mRNA in brain tissues of glioma patients that may be involved with the pathogenic mechanism of glioma-related epilepsy, which we'll be looking at further in this presentation. To address this, I worked to reject the null hypothesis that glioma-related epilepsy, shortened form is GRE, does not cause alterations in brain networks. So I mentioned that this was computational research, essentially fitting into the category of using multiple databases in order to analyze a dataset from a previous study. So what I did is I formed the proteins already found in epilepsy into StringDB to map out genes and find pathways. Then I separately went through a software called KEGG Pathway to find any overlaps between the genes in StringDB and how they are involved in significant epilepsy pathways in KEGG. Then I analyzed the NCBI dataset GSE199759, which is just its ID, through the gene expression omnibus, also known as GO2R, to understand exactly how those genes were represented in individuals with epilepsy. Then going to PharmGKB to analyze various drug treatments and create an epigenetic therapy to return the significant genes to their control values. So all of this is very interconnected, and I'll be walking you through each step. The first thing that we have to do is use StringDB. In molecular biology, this is a biological database and web resource of known and predicted protein-protein interactions. So we use glioma-related epilepsy to find the most prevalent mRNAs. When we input all of the genes, it gives us as many as it can identify in this database. In this case, that's almost 300. So we take the strongest ones, which is defined by how many of the genes are prevalent in epilepsy out of all other instances of that gene. So the two strongest out of epilepsy are the GABA-RAP-L1, which has a strength of 1.48, and GRAMD1B, which has a strength of 1.44. So one of them codes for proteins, the other one is involved in cholesterol binding and transfer. On this photo, on the right, you see that there are many lines, meaning that the genes are very interconnected and have strong appearances in epilepsy. Our main point of focus is the GABA family, which are genes associated with GABA-RAP-L1, because they work with the uptake of the GABA neurotransmitter, also known as the major inhibitory neurotransmitter. Since this is so significant, we can explore the effects of the GABA-RAP terminals through KEGG pathway. So highlighted in pink is the GABA-RAP, and GABA is formed within GABAergic axon terminals and released into the synapse. It acts as GABA type A, which is a ligand-gated chloride channel to control chloride entry. Connecting this to a wider context, in previous studies, reductions of GABA-mediated inhibition have been reported in epilepsy. So with the downregulation of GABA-RAP-L1, there's less GABA-mediated inhibition, which is influenced by GABA-A. The mediation is what suppresses seizures, so with less of it to keep it down, seizures are more likely to occur. So now we have a little idea on how everything's connected. So to see a wider range of genes and how they might be affecting epilepsy, whether upregulated or downregulated, we can use GO2R. This dataset used brain tissues of 25 glioma patients with or without epilepsy and was obtained from both males and females aged 32 to 69. Data analysis through GO2R provided me with insight into the types of genes that play a role in epilepsy and whether they are underexpressed, and to define that, it's the word downregulated, or overexpressed, which is known as upregulated. What we see through our analysis is the gene ID and also the adjusted p-value, which tells us how significant that gene is. The lower the p-value, the higher the role that the gene plays in epilepsy. The first gene that we want to look at is on the left, P27886. These bar graphs show us the expression values of that specific gene from each sample between the epilepsy and the control. So the green is the epilepsy bars, the purple is the control. So as a whole, the expression of this gene is extremely low because it's below the 0 bar. And to explain this, I'm going to use the word downregulated, meaning that it's underexpressed. As you can see, the ones in epilepsy are much further down than the ones in the control. So our p-value for this gene is 1.67 times 10 to the negative 7, and statisticians say that anything less than 0.05 is significant. So this gene has a very low p-value, and it is very significant. This is similar for our other gene as well. So like our first gene, P31769 is also downregulated compared to the control. And I want you to keep these two in mind because both of these serve as a basis for my treatment proposal. Next, we're going to look at this volcano plot. It really just shows you the upregulation and downregulation in epilepsy as compared to the control. Anything in blue is downregulated and red is upregulated. It's on the key in the bottom left. So they call it a volcano plot because it has that shape. The peak at the bottom with all the black dots means that there's not really a significant change between the control and epilepsy, so we really don't want to worry about those dots. When you analyze genes, you want to know their function, and you may see that they're playing a role in something that you should change. So I looked into the gene families as well so we could have a little more context about them. So P27886 is part of the BCL2 family proteins and is therefore a gene regulating apoptosis. That makes sense because these samples were taken from glioma patients, which is brain cancer, and brain cancer occurs when there's little to no programmed cell death, known as apoptosis. In epilepsy, there's something called seizure-induced neuronal death. It's not programmed or on purpose, and it may destroy healthy cells instead of the non-functioning ones, which will explain why BCL2 family proteins are downregulated because they no longer serve the best purpose. P31769 is a competence protein B that is required for pre-mRNA splicing. So pre-mRNA is necessary to create the final strand of mRNA, and altered splicing can pose an increased risk of seizures. The downregulation of this gene means there's increased sensitivity for seizure onset. Now, the use of an epigenetic therapy can increase expressions of GABA2 and P31769 gene. Compared to the current treatments, this kind of therapy is much less invasive, and if used correctly, can have very positive long-term effects. Now, currently, treatments for epilepsy include, and while these may be effective, they can have harmful side effects. The first group is anti-epileptic drugs. We have phenobarbital, which has really great anti-seizure properties, but the problem is that it's highly addictive, and this is already on the market for epilepsy and seizures. The second is benzodiazepines. They reduce seizures by producing sedation. And the third is just generalized seizure medication, but it's not as effective because it's not targeted. The second thing is surgery, where you remove a small part of the brain that's causing the seizures, but this is quite invasive. So I would like to continue implementing phenobarbital because it is a drug that travels directly to the nervous system, and it increases the effects of GABA, which is able to inhibit the onset of seizures. It's important that the other drugs travel to the nervous system as well, so when you put them together, they're able to function in the same area and be more effective. The second drug I would like to use is naltrexone, which is used in addictive disorder and can offset the side effects of phenobarbital. Phenobarbital, while being highly effective, can also be highly addictive, as we mentioned, and naltrexone can be combined in order to prevent this from the beginning. Now, in order to upregulate P31769, naproxen can be used because it allows DNA to be taken up by its environment and increases the levels of this gene. This is another step to bringing the genetic levels closer to normal. So, as a conclusion, I was able to reject the null hypothesis because these genes and mRNA expressions suggest a connection to the brain's pathway leading to epilepsy based on how low the p-values were, so they were very statistically significant. These new drugs can be used to upregulate and increase neurotransmitter usage of the genes and restore them back near normal conditions through less invasive procedures for epilepsy treatment. Further research could include clinical trials to improve and refine this medication. Thank you.